Our final speaker in this session, who I'm very pleased to welcome, is Dr. Amy Wedley from the University of Liverpool, who will be talking to us about monitoring AMR E. coli or resistant E. coli in sheep and beef cattle in the UK. So Amy completed a degree in biomedical sciences. She specialised in medical microbiology. And she's also completed a PhD at Liverpool in 2012 on antimicrobial resistant E. coli and Staphylococcus species in the vet visiting dog population. Her subsequent academic work has centred around two key themes: antimicrobial resistance in food-producing animals and food-borne pathogens, such as Salmonella and Campylobacter in dairy cattle and poultry. She's a lecturer in microbiology and teaches in both the School of Biosciences and the School of Veterinary Sciences at the University of Liverpool. So very pleased to have you join us today. Over to you, Amy. We can see your slides well. Thank you.
Great, thank you very much, and thank you for the invitation to come and speak about some of the work that we've been doing at the University of Liverpool. So what I'm talking about today is a bit of work that we did where we were trying to monitor antimicrobial resistant E. coli in the UK sheep and beef cattle population. So I'm just going to quickly talk about why. So we've already mentioned the National Action Plan and how that aims to tackle antimicrobial resistance through coordinated actions, looking from a One Health perspective. So not just looking at humans, looking at the environment and looking at the animal population as well. When we think about beef, cattle and sheep, there are actually limited studies that report the prevalence of antimicrobial resistance in E. coli, using that as an indicator species. So we felt it was important to be able to get an idea of antimicrobial resistance within this population. Now, there is some information provided by the Veterinary Antimicrobial Resistance and Sales Surveillance, but this is mostly limited to clinical surveillance, and it's not necessarily representative of the population as a whole, and also can be quite small numbers as well. So it wouldn't give us a bigger picture. And if we think about the impact that antimicrobial resistance in ruminants has, it contributes to a larger ecosystem. So they are using the same environment that people are. They are grazing, and their feces is used to fertilise the land. So it could have a big impact on antimicrobial resistance in the environment as a whole. So the aims of our study were to make use of some samples that we'd already collected for another study, which I'll talk about in a second, to get an estimation of the prevalence of antimicrobial resistant E. coli, but also to explore some of the different methodologies that could be used to isolate antimicrobial resistant E. coli from fecal samples. And we focused on using a non-selective and a selective method to look at how effective they were at isolating E. coli. So the samples that we used were collected from 31 farms, which had either sheep or beef cattle or both, and they had been systematically collected by a PhD student who was looking at Mycobacterium avium paratuberculosis, so another bacteria that is important in animals. And these samples were as follows. So 14 farms had both sheep and beef cattle, 14 had only sheep, and 3 had just beef cattle. And 10 fecal samples were collected from each farm. The samples had been collected between August and December in 2020, and they'd been stored at -80 at the University of Liverpool. And we then took those samples, defrosted them, and processed them for antimicrobial resistant E. coli between March and October 2022. The way that we did that was by putting 1 gram of each sample into 9 millilitres of bufferpeptone water, and then we used our 2 methods. One was just simply screening the enriched broth. So once it had been cultured at 37 degrees overnight, we plated out onto a selective media, so a differential media that allows us to easily identify E. coli, and that didn't have any antibiotics in it. We also plated it out onto a plate that contained cefotaxime, and then our selective method was to create a lawn of bacteria on a plate and then place specific antimicrobial discs onto that plate to be able to target certain antimicrobial resistances. Any isolates that we collected were then subjected to antimicrobial susceptibility testing. And any isolates that were confirmed as carrying extended spectrum beta-lactamases or potentially having AMP-C beta-lactamases were confirmed as being E. coli using mass spectrometry and MALDI-TOF. It's a way of identifying species very quickly. Here you can see the panel of antimicrobials that we used and the breakpoint, so what diameter we measured were determined as resistant. So we measured the zone, recorded it, and used CLSI guidelines and UCAS breakpoints to determine resistance. We did end up with a number of isolates, so to make sure that we didn't have representation multiple times, if an isolate had a resistant profile that was identical to another isolate that had been collected from the same sample, we would only count one of those isolates in our analysis. And our definition of multi-drug resistance is non-susceptibility to at least one agent in 3 or more antimicrobial categories. So that means that if an antibiotic was resistant to just ampicillin and augmentin, for example, it would just be resistant to one category. We also wanted to try and get an idea of the prevalence of 3rd generation cephalosporin resistance. So any isolates that had come from the plates that contained cefotaxime, or any isolate that was resistant to cefodoxime in our antimicrobial susceptibility testing panel were subjected to the mass ESBL detection double disc test. And what this does is you have discs that contain a 3rd generation cephalosporin, and then you have discs that contain that cephalosporin and clavulanic acid, which can inactivate the enzymes that cause resistance. And in the presence of an ESBL, you would see a difference of more than 5 millimeters between the two disc pairs. If there were isolates that didn't have that difference, but there was resistance, then we would suspect that they might carry AMP-C beta-lactamase as well, or instead of ESBLs. So I'm going to move on to the method, to the results. Firstly, we're just going to briefly talk about the comparison of the methods. So this table explains the number of isolates that we were able to isolate and identify from the different methods that we used. So the first column is all of the isolates that we collected, whether or not they were resistant or not. And then next one is any resistance, then looking at any MDR any MDR isolates. The first row is looking at the non-selective, so just the plates that had no antibiotics placed on them or contained no antibiotics. And then the second one, the Bartholinian method, is where you place the discs on the plate, and then we have our cefotaxime plate. And what we can see here is that none of these methods actually was 100% sensitive at isolating all of the isolates that we could potentially get. The non-selective plate only isolated 156 resistant isolates, whereas the Bartholini method isolated 668. An additional 44 were isolated from the cefotaxime plate. And thinking about the profiles of those isolates, only 71 were able to be identified through both methods. And if we look at MDR, we can see that only 18 of the multi-drug resistant isolates were actually identified by the non-selective method, whereas 209 were identified by Bartoloni. And if we look at the sensitivity, this is based on if all of these results were combined to get total isolates resistant. Bartoloni is 78% sensitive, whereas the non-selective method is 24% sensitive. And that is even lower when we consider just the multi-drug resistant isolates. So both of the methods miss a higher number of non-multi-drug resistant isolates. And the selective method, what we found was that they were better at identifying multi-drug resistant isolates and actually better at examining diversity within those isolates as well. Now, moving on to the results of resistance prevalence. So there were 31 farms, as I said. We used 442 samples, of which 278 were sheep, 164 were beef, and 1,392 isolates in total were collected, of which 869 were from sheep and 869 were from sheep, and 460 were from beef. I apologise, there's a typo. This graph here shows us the prevalence of antimicrobial resistant E. coli on the farms. So if any of the samples had an E. coli that was resistant to ampicillin or to a antibiotic, it would be classed as positive on this. So what we can see is that every farm had at least one isolate with ampicillin resistance and tetracycline and potentiated sulfonamides. And a high percentage of them had at least one isolate with multi-drug resistant E. coli present. Now, if we look at a sample level, what we can see is, in general, the prevalence of resistance was higher to ampicillin, tetracycline, and potentiated sulfonamides. When you compare sheep to cattle, there was a higher prevalence in sheep, which is the graph on the left-hand side, compared to that in cattle. And this is also true for multi-drug resistance. So there was a higher prevalence of multi-drug resistance in the sheep samples compared to the cattle samples. There were comparable prevalences of multi-drug resistance in both samples, in both types of sample. And it is important just to remember that this represents a number of samples that were tested. If we look at the isolate level, so this is every isolate that was collected from all of the samples, which equated to 1,329. So you could have a number of isolates representing each sample, and we're looking at each individual prevalent profile. It is following the same pattern as the samples. So ampicillin, tetracycline, and potentiated sulfonamide resistance is higher than all of the other ones. Multi-drug resistance is around 20%, and resistance to third-generation cephalosporins is quite low, as is resistance to fluoroquinolones, the ciprofloxacin. If we look at the resistance profiles of the isolates, one thing that we could see was quite common is that those three antimicrobials, tetracycline, ampicillin, potentiated sulfonamides, were quite common in most of the higher frequency profiles. So tetracycline resistance alone was in 20.8% of the isolates. Ampicillin and tetracycline was present in 16.3%, and then we can see variations of those represented in these isolates here. So what this is showing is that these three antibiotics are the most commonly prevalent in terms of resistant, and we are seeing a little bit of cefpodoxime resistance represented as well. If we think about some of the limitations of this study, The samples were stored for quite a long time, so between 12 and 18 months. So this could potentially have affected our ability to recover some of the E. coli that were present when the sample was initially collected. The samples had been collected to target a different organism, so we may have chosen a slightly different sampling strategy. We may have collected extra metadata for the samples. For example, we didn't collect any antimicrobial use data for these farms, which obviously would have been useful for the purposes of this part of the study. Antimicrobial use is higher at certain times of the year in these populations, depending on lambing, for example. And the fact that we only used a single time point means that we might not have been able to capture this variation as easily. So in conclusion, we looked at different methods that we could use, and that very importantly impacts the observed prevalence that we had, and that might be something that people would want to consider when they're deciding how they're going to screen samples. So one thing that you might want to consider about that is how logistically easy it is to do one method over another, thinking about the sensitivity of the detection that you're wanting to try and aim for, what sampling unit you're going to use. So are we going to work at the sample level or look at the isolate level or the farm level? And that might need to mean that you want to think about sample size in terms of the ability to actually work through all the samples that you were to collect. But selective sampling, so using a targeted approach using antibiotic resistant discs, increase the sensitivity. And actually, it might be a better way to actually use both methods. The study provides recent data on antimicrobial resistant prevalence in sheep and cattle, which is very important in improving our understanding. And there were a high prevalence of resistance to antimicrobials that are commonly used in sheep and beef, such as ampicillin, tetracycline, potentiated sulfonamides. There was a low prevalence to third-generation cephalosporins and third fluoroquinolones, which are considered the HPCIAs. In terms of what we plan to do next, we would like to do some analysis on whether or not there is an effect on whether or not there was just a single species on the farm or whether the farm had cattle and sheep. And we are currently undertaking molecular characterization of those isolates that were ESBL or AMP-C beta-lactamase positive. We're doing some PCRs to try and ascertain what specific genes are present, and we have secured some funding to do some whole genome sequencing on a subset of those isolates as well. So that just leaves me to say thank you very much to the VMD for funding this the processing of the samples, and those on the left-hand side were people who were involved in the study. Thank you.